Okay, so now we'll focus on a different area. This area is, I shouldn't say more exciting, but certainly as exciting. So we'll start with the case. The idea that the metabolome of the microbiome, and John said, here's the topic he gave me, John Trope. He said it's not that complicated. Well, I'll venture to say it's probably more complicated than I ever suspected, and so it's sort of a joke. I think tongue in cheek is not that it's very complicated. My PhD work is in biochemistry, and I tell you, I literally sit and try and figure out some of this pathology and some of these things. So it's very complicated. So let's start with a case here, a case which I took care of a couple months ago. Okay, so it's it's kind of like the microbiome. It's the good, the bad, and the ugly. I mean, many cases we see good things from it, sometimes some moderately bad things, and sometimes some really bad things are produced by the microbiome. And the metabolome becomes a pathogenic or so-called pathobiome, as some people have said. This is a 23-year-old female, previously healthy, presents to the ED after 5 days, status post having a term cesarean section. She went premature rupture of membranes, induction of anesthesia after 16 hours of membrane rupture. So she had prolonged membrane rupture, so they took her to C-section. She did fine, was discharged on post-op day 3 with her new baby. She received 1 dose of Cipro because she was allergic to penicillin, so she received in that perioperative period 1 dose of Cipro, single dose, okay? So day 5 now, she presents to the emergency department with the cramping abdominal pain, malaise, low urine output, watery diarrhea times 7 days. That's how she presents. Luckily, the intern and they sent off a bunch of labs, So temperature's 37, heart rate's a little up, not bad. Blood pressure's a little soft, but not bad. Room air, 96% sat, so her lungs are fine. White count, 16,000. Creatinine's 1.1. So reasonable postpartum, 5 days still. Stool samples, including C. diff, was sent off because of 5 days of diarrhea, single dose. The intern was very meticulous, sent it off. So she's given 2 liters of fluid. She felt much better. Heart rate came down, blood pressure got better. She was able to urinate and she ate some crackers, and she was insistent to go home with her baby. She got a new baby at home. She got, I'm not staying in the hospital, but she really didn't meet criteria for admission anyway. They called OB. OB said, look, as long as she can eat something and is drinking water, she can go home. Next day, 24 hours later, C. diff comes back positive. They called the patient to say that C. diff was positive and they needed to treat it. And her husband tells them that she is really sick today. She can't really get out of bed. She's very weak, looking sick. So they said, bring her back to the hospital. So she comes back, literally can't get out of the chair, and pulls up to the ED. They put her in the chair, roll her in a wheelchair. She's admitted from the emergency department, and the working diagnosis was endometrial infection. She had 16 hours of ruptured membranes. And they thought, well, she got endometriosis, or, endometritis, from prolonged rupture of membranes, and maybe some few products of conception. So they thought, well, let's go ahead and talk to OB. But let's get a CT just to make sure. Here's your CT scan. So if you're not familiar looking at CT scans, that's the colon. That light you see, the light area, that's the mucosal lining inflamed because it took up more contrast. The colon normally is, the whole colon is just as thick as just that mucosa lining. So it's significantly swollen, okay, dramatically bigger. Look at that, there's your uterus there, but that's colon, descending colon there, massively, thickened wall, dramatic. This is a healthy 23-year-old. Okay, so when that happens, they realize this is not endometritis from prolonged rupture. This is C. diff because you get inflamed colon. Okay, you might say it's inflammatory bowel disease, but not acute onset like this. So classic positive C. diff. She got C. diff sepsis. So they call. It's interesting, in teaching hospitals, we're a big teaching hospital. Usually goes through the chain of command. Intern calls an intern from one service. He calls a junior resident. They call, and if it's really sick, eventually gets to the attending. But this case, the attending in the ICU calls me direct. When you get attending to attending call, you got to, what's going on? Let me come, I'll be right over. So I walk over to the ICU and I see this young woman on 3 pressors now to maintain her blood pressure, norepi, vasopressin, okay, getting ready to add a third. She's in Trendelenburg. They're going, they're having a hard time keeping her pressure. She got bad C. diff sepsis. Okay, my chief resident, I drug along with me. And he says to me, Dr. Martindale, she is too sick to operate on. We got to stabilize her, resuscitate her. I go, look, she's got massive ongoing resuscitation. She's got 2 pressors on board, getting ready to start a 3rd, and we still not maintaining her pressure. My response is she's too sick not to operate on. So there's a 23-year-old healthy female being taken to the operating room now, sick as a dog, literally dying, unconscious. Her husband signed the forms. She was not awake to sign the forms. Okay, taken to the operating room, look, on 3 pressors, the mean arterial pressure of 50. That's your colon mucosa. That is C. diff. Those are pseudomembranes full of Clostridium difficile. Okay, there's normal colon, there's C. diff colon. That's the outside of the colon and the inside of the colon. Okay, now the question is here, now her metabolic signals have made her sick. So the metabolome in this case has become a pathologic microbiome. Okay, so it's killing her, literally killing her. So we took out her colon, and so I was very concerned. This is one of those cases you never forget after 35 years of surgery. During the case, I'm operating fast. I got a chief resident. I actually called another attendant to come help to make it even faster. But before he got there, I'm coming across the vessels of the colon, drainage. And as soon as we came across the middle colic vein, which is draining the majority of the colon, all of a sudden I hear anesthesia say, hey, what are you guys doing? Usually in trauma, when you hear anesthesia saying, what are you guys doing? That usually means get ready to start CPR. Okay, because there's something bad going on. But in this case, I look up at the monitors, I go, what's the matter? What's the problem? I see a blood pressure 140. And he goes, whatever you're doing, it's working. And so just cross-clamping the middle colic vein, draining the colon, within minutes raised her blood pressure. She no longer is getting those toxic metabolites into the portal vein, which is going to the liver, causing systemic effects. So dramatic effects. So now the question is, we have good metabolome and a bad metabolome. In this case, it's where it's hurting us. But it shows you how fast the changes in the colon structure can affect the patient's care. So the bottom line is, why then do we care about, she did very well, by the way. And went 3 hours post-op. She now was awake, off pressors, basically weaning off oxygenation. And she's awake enough to say, what's going on? I go, well, we just took out your colon, And she goes, what do you mean? And she goes, and what is this? The ostomy. I go, well, it's sort of temporary. We left about this much. We're going to hook you back up someday. So the question is then, what should we do about these bacteria? That sets the stage. We've sort of evolved around bacteria. They're all around us. This little guy here certainly shows us that. There's about 3 to 6 pounds of bacteria on us. And these bacteria have numerous beneficial metabolites and potential toxic metabolites, as I just mentioned. And if we look at those metabolites, short-chain fatty acids, production of vitamin K, some amino acid, neurotransmitters, we know that D-serine is only produced in the human colon. It's produced by the bacteria. D-serine is a neurotransmitter in our brain. So when we have gnotobiotic animals, we grow them without bacteria, they don't produce normal neurotransmitters in their brain. So I think we know well that there's a big connection. I've been doing nutrition for 45 years, And when we used to say we gave everybody parenteral nutrition in the ICU because we thought we knew better. We thought the more purified, the more elemental we give these people. So we'd mix it up. We'd take some amino acids off the shelf. We'd put them in a TPN bag and think that was the way we should feed our sick people. What are we doing now in the ICU? We are literally got protocols now grinding up organic foods and giving it to the sickest patients. We have 2 protocols now in our ICU. We've got 105 ICU beds at our hospital, big teaching hospital. And in that, about 30 of those patients on protocol are getting organic food as opposed to these specialized formulas. Because I think we've learned that we went too far with thinking by purifying, getting chemicals down to the raw nature of amino acids, carbohydrates, and fats, was probably the wrong way. We should be giving them as intact protein, intact foods. And I think that's what we've learned. So because what's the main factor there? We maintain a healthy microbiome. So I think that's what the change has been. So I know it sounds a little quackish, but believe me, it works. We've gone completely to this idea of supporting our microbiome, inhibiting production of toxic metabolites. Well, the whole microbiome concept is not new. I'm sure most of you realize, and I won't go into it in detail. It really started by Metchnikoff here in 1906. He wrote the first major paper talking about microbiome. He didn't call it microbiome, the bacteria changing longevity. He won the Nobel Prize for discovery of macrophage function, actually in 1904, and then used that money to travel the world to look at why some groups of people in the world live longer than others. And after he studied the Yucatan Peninsula and what's now North Korea and various places around the world, those small groups of people that used to live about 20 years longer than other people in the area. And he said the only thing that's consistent is these people have some source of lactobacillus every day. In North Korea, it was kimchi. In Yucatan Peninsula, it was llama milk. You know, in Bulgaria, it was cow's milk, fermented cow's milk, lactobacillus. So Lactobacillus bulgaricus was actually named by Metchnikoff. So he's sort of the father. We know the surface area of the gut is about the area of a tennis court, about what this room is. When we spread out the villi, the microvilli, and the little microscopic villi on top of those. So you can imagine that surface area is now exposed to that bacterial potential. 8 million genes in the bacterial genome and about 20,000 human genome. So those should be added as part of our genome. You know, so we know it's very critical. I think one of the most telling stories is this right here. 13 to 15% of the sugar in breast milk is not absorbed by the baby. So why would that be? Why would Mother Nature spend so much time to get 15% of the sugars, the carbohydrates in breast milk, the baby can't even absorb? So why is that? Because it's for the microbiome. Because even today, as I do a lot of missionary work overseas, even today, the number one killer of children in the world in the first 3 months of life is diarrheal diseases. In Bangladesh, I see it routinely. We now add a prebiotic to the kids in Bangladesh on an NIH project and a World Health Organization project. Because we know maintaining a healthy microbiome is the answer to many of this. So Mother Nature has done it for us. It's given us a healthy microbiome, a prebiotic, which makes for a healthy microbiome and saves these kids getting infectious diarrhea. So the microbiome term was coined really by Joshua Lederberg, another Nobel Prize winner. You know, he really described a lot of stuff on how bacteria share genetic material. But he coined it. He said, you know, really when we're born, 95% of our genome is absent. It's in our gut. And we rapidly recolonize that gut. And we've seen everything change. What's happened in the last 100 years? You know, changes sedentary lifestyle, we got more C-sections, domesticated pets, refrigeration, and on and on. But the main one is what's happened to our food intake. You know, dramatic changes, fats, proteins, emulsifiers, sweeteners, antioxidants, preservatives. This was nicely shown us by Dr. Reed in the last lecture. So if we look at infectious disease, what's happened? Look at infectious disease, we've done great. TB, rheumatoid, hepatitis A, mumps, all coming down in 2000, you can see. But look what's happened in inflammatory diseases. All these multiple sclerosis, Crohn's, asthma, type 1 diabetes, all these are inflammatory diseases. Is part of that because we've changed the microbiome? You know, so what's the cell signal that allowed this change to take place? It's dramatic. We know adipose tissue, liver, all these things influenced by the gut microbiome to see end result effects. Every organ system, that's too small to see, but this one you can see, you know, short-chain fatty acids, TMA, bile acids. Look at this. These stimulate hormone secretion, which affect virtually every organ. Our ability to take protein up in the muscle, the ability of the liver to metabolize fat. All these things are taken up, and routinely altered by the microbiome metabolome. Well, we talked about epigenetics. John hit epigenetics fine. But look at the number of papers on epigenetics. You know, we didn't talk about this very much until really about 2000, and then it took off. And this is not just, this is a log scale, okay? So this is huge number of papers now. Discussion, and you know, we won't go into this because the next couple lectures you're going to see more on epigenetics. But basically, epigenetics in my mind, again, I'm not a geneticist, so epigenetics to me is a new area. I do some biochemistry, but so I can partially understand. But it takes a lot of science for me to figure this out. But you know, it helps explain the relationship between individual genotypes and the environment during all stages of life. And it's a mechanism which integrates the environmental changes at the cellular level, enabling the cellular plasticity. So methylation, acetylation, phosphorylation, etc., those changes to the genome, which are not translational changes, they don't change the DNA, but they changed these post-translational effects on the DNA, okay? And significant. So what do we see? What metabolites are in the gut? Vitamins, short-chain fatty acids, polyamines, polyphenols, all these non-absorbed digested carbohydrates. So we see dramatically, and we see all of these things affected. Again, I won't bore you with the details, but again, you'll have these in your slides. But show, like for example, DNA methylation is usually associated with suppression of gene transcription, okay? So histone methylation usually transcribes and activates transcription. Okay, so you can see that these changes are not, these are post-translational changes. They don't change the DNA structure itself. They are post-translational effects which turn on or turn off genes for us. Thereby, we see that. And lots of now data which you saw this morning already on microRNA, changing these lipid mediators as Justali showed us. And again, so John's saying it's not so complicated. Well, I think it's the exact opposite. Look at it. When you look at this is the microbiome, this is polyphenols, vitamins, they all have interactions here. And again, I can't really read that too much, but to show you, it's very complex. Very complex. So let's look just at the short-chain fatty acids, because they're probably the one that get the most press when we talk about these metabolome. And we look at short-chain fatty acids, and the big player is butyrate. 4 carbons, small molecules, rapidly produced by the cell, rapidly taken up, until, you know, we used to think, when I first started hearing about butyrate in the 80s, we talked about butyrate being the primary fuel for the colonocyte. We thought if the colonocyte can take up any fuel it can, it will use short-chain fatty acids for the fuel. Well, then we found out that, guess what? The myocardium will preferentially use short-chain fatty acids. And now we knew most tissues will preferentially use short-chain fatty acids, in fact. They'll take them up and use them. And so when we started talking about, well, how do we translate what the bacteria are producing in the gut, in the colon, short-chain fatty acids, usually 80 micromole, how do we get to the serum? We didn't know about a translator in the 80s. Not until 1991 did we actually describe the transporter. Dr. Ganapathy and Leibach described the transporter for short-chain fatty acids present in the colon. We had them in the kidney. We'd seen them in the kidney, but not till 91 did they actually describe the transport in the colon. So that gave us the mechanism now that butyrate produced in the colon can have a systemic effect, not just at the colonocyte level. So a systemic effect. And we know that, we know they activate G proteins, as they showed us earlier. Regulates transepithelial fluid transport, reduces mucosal inflammation, and we can read the list. When we talk about cancer development, histone deacetylase. That's a big issue of anti-cancer effects affecting histone deacetylase. So I think we've got lots of data. We know butyrate affects mitochondrial activity. Look at Alzheimer's, Huntington's chorea, all these CNS effects. This is just the brain, G protein receptors, histone acetylation. So we see multiple effects in that case. Now, when we look at the cell, what's going on in the mucosa? We know that in the crypt, as you know, in the crypt, there are all these little crypts in the GI tract, these folds. We know that, in fact, butyrate production, butyrate is about 5 to 8 millimoles here and about 0.5 millimoles here. So as we go down the crypt, the concentration of butyrate goes down. Up here where the bacteria are, so the butyrate production is. So we know the cells are influenced by this. This is the cancer cell here, for example. We know the butyrate affects that cancer cell by downregulating the ability to produce that cancer. Lots of data now on histone deacetylase. So very interesting data. So we know the microbiome affects virtually everything we do affects the microbiome, whether just traveling across the country. We know age affects it. We change over time. The species change over time as we go, as we grow older. We know there's actually 4 studies now showing just getting on a plane in the United States and getting off the plane in Asia, 12 or 14-hour flight affects your microbiome. Just 14 hours, we see a dramatic change. Okay, getting a domesticated pets, it's very interesting when they follow the microbiome of the families that get a new pet. It's very interesting. The youngest child has the most rapid change, and I think it's because of this. Guess what? The dog's cleaning up after the kid from eating, So there's no question, that we rapidly take on the microbiome of our pets, and the pets take on our microbiome very rapidly. And it changes very rapidly. It's probably good for us, So, that's how fast it changes. We know taking a single broad-spectrum antibiotic, we go from about 1,000 species, plus or minus a couple hundred, down to about 200 species within 48 hours. Single broad-spectrum antibiotic. So what happens to when you get all these people get surgery and all this stuff? We're altering our microbiome. We need to get it back to normal as fast as we can. And we know that, the medical community as an MD, most doctors think, oh, this is quackery. Come on, this is just not science. And we know, in fact, it is science now, but it's taken forever to get them to see this as science. They think I'm a nut in my hospital, most of them. Okay? So, it took like lay populations, okay? Microbiome maketh the man, 2012. The cover of The Economist magazine. It wasn't Nature or Science, it was The Economist magazine. So the lay press and the business press was taking this on. New York Times here, Wall Street Journal, look at this, talking about microbiome changes bacteria. New England Journal, Scientific American, New York Times. And only recently, only in the last 3 or 4 years have we got great data in scientific journals, okay? Annals of Surgery, Nature, JAMA, Proceedings of the National Academy of Science. Every month now, virtually something on microbiome. And every one of these diseases, we can talk an hour about microbiome changes and epigenetic changes with every one of these. I'm a trauma surgeon, general surgeon, so I deal mostly in the general surgery critical care arena. Now, we know we communicate, right? Everything communicates. Like E.O. Wilson is sort of the father of social biology. He says, in war and nature, it's a battlefield. Make no mistake, one needs secret codes. So these bacteria, we now know, communicate with each other. We didn't think bacteria communicate. We now know they form social groups, so-called quorum sensing. When about 10 to the 4th bacteria together, they start to form a community and decide who's going to do what work, okay? We know bees can talk to each other and tell each other where the bees come from, and clearly these two are communicating, okay? And there's no written language here, but they're communicating, believe me, okay? So if we look at that metabolomics, then it's a scientific study of the chemical process involving metabolites and basically communications within cells. The question then becomes, does altering the microbiome make a big difference? Well, here's a great example. This is work by John Alverdi's group, okay? Showing here very nicely, you'll see red color starting to develop here, okay? That's expressing pathogenic features, okay? It takes very little to cause a normal microbiome to become non-pathogenic to become a pathogen. This is Pseudomonas aeruginosa. The only difference here is this one is exposed to serum from the patient who got an opioid. This one is the vehicle only. So the serum of the patient getting opioids allowed expression of virulence genes in this Pseudomonas. We know that very well now. Just giving narcotics, giving broad-spectrum antibiotics allows virulent gene expression. Okay, so what we do has an effect, so-called changing of the microbiome to a pathobiome. Now, I don't want to bore you with the details of the study, but this next study this is the study here, published in 2013. A very important study because I think it shows us how things change. So what they did here was they did 24 hours. They did lethal Pseudomonas morphotype develops, okay? What they showed here was they took a sham, this is a rabbit model. They do an operation. They open the belly and close the belly, okay? And then first they put in this Pseudomonas, benign Pseudomonas, same substance in our colon. They put the colon in, do a sham operation. 24 hours later, they isolate out the bacteria from the sham-operated animal. Okay, here they do a hepatectomy on a liver, on a rabbit. Basically, left lobe's gone. They take half the liver out. Okay, 24 do the same thing. After that, they put in the bacteria, do the sham operation, spin down that, and break it up. They give half to the sham, half to a hepatectomy of the normal bacteria. And look at this. The animals that got a hepatectomy, 100% fatal. The animals that got a sham operation, 100% survived. So what happened? The only difference here was one got bacteria that was in an animal that had an operation, just a sham, one got a hepatectomy. So the bacteria changed from non-pathogenic to pathogenic, enough to kill the animal. So that's what we've got to prevent. We've got to prevent these transitions and changes with all these dramatic things we do. That's just the beginning. We know that people, if they've had prior antibiotics and come back into the hospital within 3 months of having an antibiotic, their mortality from sepsis is higher. Look at this, across every range here. This is Apache score, severity of illness. Almost doubles your, if you got an antibiotic in the last 3 months, you come back in, guess what? You've got to double the chance of dying from the same disease. So we've altered the microbiome. You know, our fear of bacteria has gone crazy, right? I mean, ever sterilizing them? They go to the grocery store now, they got these wipes. You got to wipe everything off. And give me a break. You know, how do you develop your immune system? We're going to kill all the bacteria? That's not the answer. We can't kill them. We got bacteria now that are resistant to every antibiotic we have. We thought we're smarter. Oh, the scientists will develop new antibiotics. Guess what? We got bacteria now that we have no antibiotics to cover, and they're coming more and more. We're not getting new antibiotics. So the idea that we're going to kill the bacteria, like this fella here, you know, we're not going to do it. Remember that mucosa protects us. We talked about that already. And there's multiple mechanisms. Competition, let's talk about it. We know now the cellular level. This is cell membrane. We know if you got lactobacillus, you got certain lactobacillus species out here, we get decreased production of anti-kappa B getting into the nucleus and multiple other steps. So all these mechanisms, we're not going to bore you again with the details, but here, a good example, Lactobacillus rudei inhibiting H. pylori, Lactobacillus rudei inhibiting Staph aureus. So bacteria in our colons can inhibit these. What about enhancing heat shock protein? Remember, heat shock protein is the protein which is expressed by the cell to protect it from subsequent stress. We know in the presence of the right bacteria, the signal is increase heat shock protein to protect you from future stress. How about blood flow? To heal any GI tract lesion, we've got a good blood flow. As you can see here, this is a germ-free animal. Conventional bacteria increases about 60-fold blood flow. This is almost 100-fold blood flow with Bifidobacterium theta. So the right bacteria, again, enhance blood flow. So we're now looking at picking the right bacteria or giving the right prebiotic to stimulate blood flow and healing for different diseases, with things we need to do in the GI tract. How about immune function? We know now, this is the animal model where we can give an injection. We can show that given the right probiotic, in this case, or prebiotic, we can actually enhance the lymphoid tissue and the T cell response. Now we have a clinical connection. Giving the correct bacterial profile, we actually, given before immunization, a better response to to the immunization. So given elderly people over 65 prior to the immunization, given the right prebiotic or probiotic, actually we increase our immunoglobulin production. And here's the key: no increase in inflammation. Short-chain fatty acids, again, just Ollie talked about that. This happens to be a macrophage, scanning electron micrograph of macrophage, attacking a poor defenseless bacteria, okay? To do that, what has to happen? First, it has to recognize that bacteria is there, that macrophage has to recognize it's there, and then it sends out an oxidative burst, right? It sends out, a pseudopod. This is a pseudopod. It engulfs that bacteria and then produces an oxidative burst to kill that bacteria. We know the average macrophage kills about 7 to 9 bacteria before it undergoes apoptosis. In the presence of short-chain fatty acids, we get a shift, and we can now kill up to 15 bacteria before it undergoes apoptosis. So in the presence of short-chain fatty acids, a metabolite of the microbiome, we see activation of the killing ability of macrophages. So I think very exciting data. We know that these are chemical mimicry. Just recently published in December of 2017, G protein coupled receptors made by bacteria to activate these receptors. Okay, G protein coupled receptors we talked about last time. And here's some very new data, just again published in December of 2017, where we show that short-chain fatty acids actually enhances muscle function and mitochondrial biogenesis. And that the combination of a good combination of prebiotics actually increases muscle uptake and muscle function. So this is the first time we've actually shown that we can change the mitochondria and muscle with dietary changes, minor changes. And they showed that exercise plus these changes dramatically change the microbiome, as opposed to the guy who's sedentary, who doesn't have a good microbiome. Very interesting data. So lots of mechanisms, as I talk about, since we won't go over these, but we talked about most of them. So there's lots of mechanisms involved here, 2 whole pages of different mechanisms involved in how they work. Let's look at some of the, sort of where the rubber meets the road. Let's look at some of the clinical studies. Well, I think does changing or keeping the right microbiome on a daily basis affect outcome? This is a great study done in Lund, Sweden, in the Volvo factory. Okay? What they did here was they gave half the people in the factory a free drink if they drank their drink through a straw. Okay? And they gave the other half the same straw, but this one had no probiotic coated in the straw. So we got 300 million lactobacillus species in that call, and then they looked after 1 year, they looked at how many people missed days of work. The people who got the probiotic coated straw missed half the days of work than those who didn't. Pretty dramatic. How about kids? How about if we keep the right microbiome and signals in kids? Look at this. They're given placebo, probiotic, or Lactobacillus reuteri probiotic. You can see this is basically not functional. And this is number of clinic visits, days absent from daycare, and antibiotic prescriptions. The number of antibiotic prescriptions given to these kids after 12 weeks of supplementation goes down 65%. This is actually done in the state of New York. So we know when a kid gets in this state, if a child gets an antibiotic at school, the teacher has to write down what was given. So that data is pretty solid. So the number of antibiotic prescriptions goes down 60%, and it's held up in others. Now, what about this? You say, wait a minute. What's the signal here? How can it be that changing our microbiome signals can alter pregnancy? Something in our gut can alter the fetus, growing fetus? Come on. What's the connection there? Well, this was the first big study, gestational diabetes, control group, placebo, and probiotics. Remember, about 30% of women in a high-risk pregnancy group, this is a high-risk pregnancy, will get gestational diabetes. And they showed with keeping the right microbiome signals, they could lower it down to 13% versus control at 30%. You go, come on. In this country, high-risk pregnancy run about 33% gestational diabetes in a high-risk pregnancy group. That's what this was. That was too good to be true. That just changing the dietary regimen could do that. Well, look at this. 189 articles now on gestational diabetes. Look at this. Primary outcome, gestational diabetes, preeclampsia, all these are secondary. Look at this. Conclusion of 189 articles, we decreased gestational diabetes, decreased maternal fasting glucose, decreased preeclampsia, and CRP, moderate inflammation, is down. So this is not just a single paper out of Norway. This is now routine. We also know the kids of the mothers who had this right microbiome have less atopic disease by 40%. So, what about neonates? These are very low birth weight infants, maintaining a healthy microbiome and metabolic signals. These are 7% of kids of very low birth weight, under 1,500 grams or 3 pounds, under 3 pounds. You can see very high risk of necrotizing enterocolitis. Look what happens. We give a 5 probiotic combination and the reduction in NEC from 9% to 5%. All of our babies at our hospital now under 3 pounds, actually under 4 pounds, get probiotics to maintain. And we get them back on mother's milk immediately. What about the brain? There's going to be a couple of toxins, so we won't. How does the GI tract affect the brain? This is a big leap for people. You go, come on. What's going on in my guts affecting my brain? We get the blood-brain barrier there. It's a great filter. You know, very little gets across it that we don't want it getting across. Guess what? A lot of the metabolites from the microbiome clearly get across. We know they get across very rapidly. And again, it's too small to see. This little stressed fella here, that's a stressed mouse. They have actually got a genetically stressed mouse. When you change that stressed mouse microbiome, guess what? It becomes more calm. And the other way, you can take germ-free animals, take bacteria from the stressed mouse, and give it to a germ-free animal, and they get stressed. And we have all kinds of models for stress, you know, where they go walk out on an unprotected limb and things like that, and they check it, and they film them, and they see they won't do the things because they're nervous or too stressed out. They stay hidden in the corner. They won't experiment in their cages and things. So now we've got lots of data on anxiety, behavior, depression, learning. We're currently involved in a very big project in Laos and in Thailand, in the hill country of Thailand and northern Laos, where we're giving a whole grain microbiome stimulant basically to kids. It's a whole grain rice, and checking learning ability, cognitive ability over time. It's a very exciting study. I think will make a difference. Now, last couple slides, we already talked about C. diff in our early part of our case presentation. But look at this, just on antibiotic-associated diarrhea. In JAMA 2012, 82 randomized clinical trials, 11,000 patients, probiotics given at the time of starting decreases risk of antibiotic-associated diarrhea. C. diff, we already talked about, good data. And now the timing is key. Because remember, if you give the antibiotics a couple days and let the microbiome change, you're at risk. This is 6,000 patients. You can see here somewhere, 6,000 patients just published in November of 2017. C. diff incidence. Basically, if you start the probiotic or the right microbiome manipulation on day 1, you will not get C. diff. If you wait 48 hours, your risk is a little higher. If you wait 72 hours, it's even higher. If you wait 5 days, it's the same as in control. So really, we need to start early on maintaining microbiome. Now, we've been talking about giving prebiotics or probiotics. What happens when patients get sick? Now we're talking use of fecal transplantation, okay? Giving back stool, because it's really too simple to give one bacteria almost. It's kind of simple-minded to think we're going to change major things with only one bacteria once we've got a problem. The idea of refaunation, re-establishing normal flora in the gut, many people are giving stool transplants, okay? And we do it mainly for C. diff, but there's a couple other protocols approved by NIH under protocol where we give it for that. Now, normally a few bacteria hide in the crypts. This is a crypt, those creases, right? These are bacteria hiding in the crypts right there, okay? And then we give antibiotics, and they still stay there to refaunate what goes on. So you can see, so we end up with a few bacteria in the crypt, and we want to get back to this. Okay? So what do? And we know for C. diff, that's what we do. We know that for C. diff, within 12 to 24 hours, patients with C. diff sepsis, as long as they're not so bad we got to operate on them, if we give them a stool transplant, they get better. Literally within 24 hours, they're talking, walking, feeling better with a stool transplant. We buy the stool from Mayo Clinic. It must be a good stool from Mayo. You can get it from a family member, but you can also buy it for $250. They got 60 people at Mayo that give their stool, and they've been tested for every known disease to mankind. So we know that's pathogen-free stool. And we mix it up there like this, and we give it by colonoscopy. Usually have 250 cc's. We give 50 cc's at 5 points along or we give it by NG tube. Most people, they're too sick to say yes or no, so we give it by NG tube. Okay. So what about sepsis? If you look when they give it for sepsis, end-stage sepsis, refractory sepsis. Look at this. Within 2 days recovered. These people had all antibiotics already, severe sepsis. Within 24 to 48 hours got better. So treating refractory sepsis is now. But can microbiome manipulation change this? No question. And we've got data to show that. And I know if we take stool from a thin person and a heavy person, give it to a notobiotic mouse, we can make them thin or heavy. So the bacteria make those changes. And I won't bore you with the details of all this. We can do it with prebiotics, as I mentioned too, whole grain issues. And I want to talk about only this thing real quick. Giving prebiotics on a daily basis lowers all-cause mortality. There is no question now. Okay? Look at this study here. 780, basically 750,000 people, this published in Circulation in June, all-cause mortality decrease. Again here, British Medical Journal, high-quality journal, June of 45 studies, minimal 3 servings a day, basically 3 ounces a day, all-cause mortality decreased. So it doesn't take much. So eat a wide variety of foods, daily intake good probiotics, be cautious and watch out for the claims. Because remember, you got an area where a lot of people get kind of crazy claims. The future looks fantastic in this area. We're looking now at various diseases and disorders, even using these sort of concept on intra-abdominal and non-cancer things. So I think the days of killing bacteria are gone. We now have to think about bioecological control. That's what we should be focusing on, not killing them. Okay, thank you very much.